Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem, that's me. Today we will talk about Lamda Phage genome organization and its features. As you all know that Lambda Phage is very complex virus and also it is very extensively studied bacterial virus. And the size of the genome is 48.5 kb, roughly 49 kb. And when they isolated the genome from the Lambda phage, it's shown that it is a linear double-stranded DNA molecule. The physical map of Lambda phage reflects the various important genes involved in the induction of lytic or lysogenic cycles, replication of DNA, synthesis of capsid, tail fibers, collar, and post lysis, and assembly, and so on. The most important feature of the Lambda phage genome is clustering. Clustering means the genes which are related to the one metabolic function, they're packed or they placed at one place. That is called as a clustering. So you can see there are 4 clusters you can see in the Lambda phage genome. Those are related to the synthesis of capsid, head, and assembly, and non-essential region, and the genes responsible for integration and recombination, DNA synthesis, and host lysis. So we will go through them by one. You can see here the left side border, close to the left side border, this A, W, B, C, D, E, F, Z, U, V, G, T, H, M, L, K, I, J. So these are the genes which are responsible for synthesis of capsid, or you say head, tail, and assembly. And the next region, this region is almost there between 0 kb to 18 kb. And the next region is the B2 region. This B2 region is not essential for cell viability or replication. If you can delete this region, right? If you can delete this region, it's not going to affect the Lambda phage viability or replication. That's why it is called as a B2 region. Then this corresponds to from 20 kb to 28 kb. Then immediately after that, there are genes which are responsible for a recombination, means integration of the genome, Lambda phage genome into the bacterial chromosome during the lysogenic cycle. You can see the int, cis, and exo, which are there between 28 kb to 35 kb. Then C2, N, C1, and Cro. These are the genes which are responsible for early regulation. They are present after the genes which are responsible for integration, excision, and recombination. Then OP. These are the genes which are there at 42 kb to 44 kb. They're at that place, and they are responsible for DNA replication. And the Q is a anti-terminator. And the SR. These are the genes which are responsible for post lysis and release of the phage progeny. And finally, you can see there are 2 cohesive or complementary single strands are there at the pi prime region of the linear DNA molecule, they're called as cos sites. Cos sites. So the lambda phage DNA have single-stranded nucleotides at the pi prime end of the DNA, and they are called as complementary. They are called as a cos sites, and they are complementary to each other, whose length is around 12 base pairs. You can see the composition also: CCC, GC, CG, CT, GG, AA. Yeah, that's it. So these are the cos sites which are there at the 5' end of the lambda phage genome. Once again, we'll see these genes function in detail. And as I told you that the B2 region, which is there between 20 kilo base pairs to 28 kilo base pairs, when you deleted this one, and is not going to affect the phase viability or replication. So this is very critical for the development of cloning vector. See, A, W, B, C, D, E, F, Z, U, V, G, T, H, M, L, K, I, J. These are the genes which are responsible for head, tail synthesis or biosynthesis and assembly. So you see, yeah, these are the genes which are responsible for head, tail, and collar synthesis, as well as they're also responsible for assembly of these proteins. And the next one is int, cis, exo, integration, recombination, and excision. Whenever phase undergoes the lysogenic cycle, it will integrate into the bacterial chromosome through the attachment sites. So that integration is called as a site-specific recombination. Site-specific recombination. So you see, the site-specific recombination is mediated by these genes. You see, this is a prophase, this is a bacterial DNA. So lambda DNA, when it integrates into the bacterial DNA, that is called as prophase, and the lambda DNA have attachment sites, and bacterial DNA have attachment site through which both of them are undergoes the recombination through attachment sites. This kind of recombination is called as a site-specific recombination. So when a phase is infected, then the lambda DNA is integrated to the bacterial DNA, then that bacteria is not susceptible to any kind of infection by any other virus. So that is called as a super infection or immunity. So here C2, N, and C1 and CRO, these are the responsible for early regulation, immunity, and super infection. Next, OP. As I told you that they are involved in the DNA synthesis or DNA replication. So O and P, these are the proteins which initiates the DNA replication through theta mode of replication. Theta mode of replication. So theta mode of replication is a bidirectional replication. Bidirectional replication. So then there are genes called as Q, that's a anti-terminator, RED and GAM. These are the genes which are responsible for conversion of theta mode of replication to the rolling circle replication. That's a unidirectional replication. So RED and GAM genes are responsible for conversion of theta mode of replication to rolling circle replication. That's nothing but sigma replication. Then lysis of the host is mediated by the two genes called as SR. So when progeny, when phage particles are synthesized and assembled, and they break down the cell membrane as well as the cell wall through the autolysins. They are called as lysins. So they are endolysins and Holins molecules. So the Holin molecules forms the force in the plasma membrane through which lysine molecules enters. They're also called as muramidase and break down the cell wall and then releases the phage particles. So S and R genes which are responsible for the lysis of the host. You see, release of the phage particles. Then coming to the last one, cause sites. As I told you that cause sites, which are there at the pi prime end of a linear DNA molecule, and those length is 12 base pairs, and they're complementary. Whenever phage injects its DNA into the host, that's a bacteria, then the linear phage DNA immediately circularized by using the cause sites. By using the cause sites, they're nothing but complementary sites. So cause sites are useful in the circularization of the lambda genome in the host. You see, this is a linear one, and these are the cause sites, and it gets circularized by using the cause sites. So circularization of the genome is very, very essential for two things. One is that to undergo the lysogenic cycle, the linear one has to convert into the circular genome, then circular genome of the phage, and the circular genome of the bacteria undergoes the recombination and enters the lysogenic cycle. If not, the circularized phage DNA also undergoes the replication to induce the lytic cycle. Right? So this is all about lambda phage genome organization and their features. And if at all you see the material is useful, try to subscribe my channel. That's Dr. K. PREM-PRIMER. And if you want to share, there is no restriction. You can share it. And if you like it, try to give thumbs up. And if you have any comments or clarification or doubts, write to me. I'll get back to you once I see your comment. Thank you all for listening. Have a great day. And I forgot to tell you, wish you a happy and healthy, happy new year. See you all. Until then, bye-bye.